thank you for joining us this Friday lunchtime. My name is Hugh Brady and I'm the Vice Chancellor and President of the University of Bristol. Welcome to the final of this year's 3-minute thesis competition. For those of you unfamiliar with the competition, 3-minute thesis challenges our doctoral candidates to deliver a compelling 3-minute presentation on their research topic and its significance. In doing so, they're permitted to use just one supporting slide. Now, traditionally, this event is the highlight of our annual Research Without Borders Festival of Postgraduate Research, and our participants present in front of a judging panel and live public audience. However, due to current public health guidance, we've had to take a slightly different approach this year. Our finalists have already delivered their presentations to the judging panel. The panel has deliberated and their decision will be announced at the conclusion of today's broadcast. Our winning finalists will then have the opportunity to represent Bristol in the national 3-minute thesis semi-finals. Now, of course, you, our audience, will still have the opportunity to take part in today's proceedings by voting in the People's Choice category. So we'll shortly be hearing presentations from 6 of our fantastic doctoral students. They're part of a community of more than 3,000 research students at Bristol. Many of them are making a valuable contribution to local, national and global efforts to tackle COVID-19 and its associated challenges. I'd just like to take the opportunity to thank and celebrate each and every one of them. You're an integral part of our university community and we can all be proud of your achievements. So thank you again for joining us this afternoon and best of luck to all our finalists.
Do you remember your life before coronavirus outbreak? Probably you were in a coffee shop with your friends talking that you want to go to Australia to go scuba diving because you love fish. But at the same time, you were concerned about global warming and you know taking an airplane means greenhouse gas emissions. What if I tell you that airplanes could save fuel consumption by mimicking your favorite fish and reducing the coffee grains? Avians, like hawks and eagles, can fly silently and efficiently because they have flexible wings that allows them to change the wing shape in a smooth and continuous way to keep the airflow attached. That's why it can reduce drag and noise. In contrast, aircraft wings are very stiff. They cannot change the wing shape. That's why they need control surface like ailerons that aggressively separates the wind flow, leading to a substantial increase in drag, noise, and fuel consumption. On the other hand, I am working with a flexible wing inspired by the fish skeleton. It can change the wing shape in a smooth and continuous way to keep the airflow attached, exactly like avian wings. That's why it can reduce drag, noise, and fuel consumption. On the other hand, flexible wings lack something. They are too floppy to hold and lock the optimal shape. In other words, I got the fish skeleton to change shape, but I am missing the muscles to lock and hold the optimal shape. So how do I get the muscles? Well, have you ever held a vacuum packed ground coffee? It looks like and feels like a brick. Try to bend it and you will realize that it's so strong. But why it's so strong? Well, it is the vacuum. The vacuum squeezes and locks the grains together, make it into a solid brick. But if you open the bag, the vacuum is released, the grains are loose, and the whole structure behaves like a fluid. We can mimic soft and stiff muscles by using vacuum packed particles. My flexible wing has empty chambers along the spine. If I fill it with coffee grains, I have a new concept that can switch the stiffness by playing with the vacuum. So when the vacuum is off, it can change the wing shape. And when the vacuum is on, it can hold and lock the optimal shape. Now I have a smart wing that can switch and shape and change the wing shape to save fuel consumption. Now you will be asking why coffee grains. Well, it's not because they are tasty. It's because they are light in weight and the shape is rough. That roughness increases the friction between the particles, creating an interlocking effect and therefore increasing the stiffness. So when coronavirus ends, and you visit a coffee shop with your friends, you will know how to make greener airplanes. So thank you very much and enjoy your coffee.
How do we make sure we're in the right place at the right time? Can you cast your minds back for a second to our world before lockdown? Imagine our cities like on the top left of this slide, constantly in motion. Traffic moving on roads to get everyone and everything where it needs to be. Like the camper van on the top right taking surfboards to the beach. Pioneering engineering efforts have led to complex transport networks. But I've got news for you. Evolution did it first. Inside every cell in our bodies, transport processes are essential to stay healthy. And whilst we might be stuck at home, inside our cells, transport goes on. As you're sat here imagining driving down roads, right now, billions of nerve cells in your brain are working hard to communicate through electrical signals and an intricate neural network. To send these messages, signaling cargo must be trafficked long distances through each nerve cell and to the next. Transport inside cells in our brain is essential to allow us to see, hear, think, feel, and move. The roads of our cells are a network of filaments called microtubules, shown in blue on the bottom left of this slide. These assemble throughout the cell and form the tracks for transport, our cellular motorways. The cars are motor proteins in red on the bottom right. These pick up their cargo and walk along the microtubule tracks to deliver them to their destinations, the Ubers of our cell. They have motor domains which act like feet and allow them to literally walk step by step along the microtubule tracks, dragging their cargo with them. If you can escape from your fantasies of unrestricted travel for a second, you might start to remember that sometimes transport goes wrong. How many times have you been delayed due to traffic from an accident or roadworks, or a cancelled bus or train or plane? Sometimes things go wrong inside the cell too. When this happens in cells in the brain, things end up in the wrong places. And eventually, that can lead to neurodegenerative diseases like Alzheimer's or Parkinson's disease. I'm researching one of these proteins to find out how it works, and in particular, how it picks up its cargo. Ultimately, if we can understand the mechanisms by which these proteins work and go wrong, that will help us to understand and treat brain diseases. I've designed a new tool which allows cargo to attach more tightly to this motor protein, increasing the amount it's transported. The position of different components in the cell depends on a delicate balance between motor proteins acting in different directions, a bit like a tug of war. And much like a really muscular teammate, our tag allows us to drive transport in one particular direction, giving us control over the position of components in the cell. I'm dreaming of a world where we can understand and control transport inside the cell to provide a biological breakdown recovery service and prevent debilitating neurodegenerative diseases. There's still a lot of work to be done to understand the amazingly complex transport networks that biology has engineered inside our cells. But by designing tools to take control over motor proteins, we're taking steps to get things moving in the right direction. Thank you.
Have you ever donated blood? From treating long-term patients with blood-based diseases to providing their life-saving means in emergency cases, blood transfusions, made possible by your donations, have become a vital component to modern medicine. In the UK, 2 million pints of blood a year are required to fulfill the medical needs of our society. To put this into context, as a nation, we consume almost 4,000 times this in pints of beer. However, unlike the brewing industry, we just simply don't collect enough blood to meet the demands of our ever-growing and aging population. So over the past decade, scientific research has instead begun to find ways in which we can grow red blood cells in the lab. This would mean that A, we wouldn't have to solely rely on your blood donations, and B, we wouldn't have to worry about screening that blood for infectious contamination. Incredibly, we are now at the stage where we can produce red blood cells outside of the body through mimicking natural processes. What starts as an unlimited pool of early blank cells called stem cells, in a glass dish, we're able to nurture and gently encourage the formation of fully functioning, mature red blood cells. Now, this is impressive stuff, But I want to go one step further. I want to be able to produce blood that not only functions to provide our bodies with the oxygen they need, but that simultaneously functions to help us fight disease. I'm exploring how these cells can be modified to help neutralize the damage caused to tissues during organ transplantations and chronic wound formation. To do this, I'm using pioneering techniques that triggers the stem cell's DNA to produce new and helpful disease-fighting cellular machinery. Now, once these cells have fully matured, they'll be stuffed full of new machinery, but naturally contain zero DNA. So this provides us with a long-lasting therapy that poses no risk of causing DNA changes anywhere in our bodies. So, perhaps akin to the super serum that made Captain America, our enhanced red blood cells will also help to keep some bad guys at bay, just inside of our bodies. I'm still in the process of trialing exactly which cellular machinery and in what combination will rear our best line of defense. So I'd hold on to your tablets for now, but look to a future of blood 2.0, lab-grown blood that combats disease, as well as delivering us oxygen.
The Nobel Prize-winning physicist Richard Feynman once said, "What I cannot create, I do not understand." This is true of many niche areas of science, not least my own. But I really quite like this quote, and I'll tell you why. I'm a protein designer by trade. Mother Nature was also a protein designer. In fact, she was the pioneer of the field. I'm very much in awe of what nature has done, but to be honest, I don't really understand it. I don't understand it because I didn't do it myself. Proteins carry out chemical processes that are essential to life, like carrying oxygen around your body and helping you digest your food and fighting off nasty infections. Proteins are built from a Lego set of 20 amino acids, like you see on the top left of the slide. These amino acids are linked together into long chains, which then fold up into 3D shapes, like you see on the top right. However, we don't fully understand how we get from amino acid chain to protein shape. This is called the protein folding problem. In my PhD, I'm trying to solve a smaller version of this problem. I want to build a new protein that can do some simple chemistry. However, this protein shape has never existed before, so there is no instruction manual or picture on the box that will show me how to make it. Thankfully, I've managed to figure out how to make the protein shape I need, and I just have a little bit more work to do before I can start installing the chemistry. On the bottom of the slide is a picture of the bullet cluster. Here, NASA found matter, shown in red, as well as dark matter, shown in blue. Now, you might be wondering, what has space got to do with proteins? Well, the matter reminds me of natural proteins, and the dark matter makes me think of the proteins that could be. We can explore proteins in the same way that we can explore space, and there are a lot of dark matter proteins to explore. The more we explore, the more we can learn about our biology and its limitations, and how to overcome those limitations. We could design and build many new proteins based on our needs. We could make new therapeutics, vaccines, materials, and sensors. Maybe we could even do some new chemistry that nature hasn't yet had the chance to explore. So what I like so much about that Richard Feynman quote that I mentioned at the start of my talk is the truth it holds. What I cannot create, I do not understand. It's true, and so is the inverse. I can create, and I will understand. Thank you.
Somewhere in the middle of the English countryside is a research facility where scientists are trying to produce clean, practically unlimited energy from a miniature sun. Inside this facility, scientists fuse or stick together molecules And this releases energy, just like in the sun. One of these molecules, which is called tritium, is radioactive. Now, don't panic. You'll receive about as much radiation from the tritium that's used in these experiments as you would from eating a banana. But that doesn't mean we don't want to know where it is and how much of it there is. My research focuses on designing a radiation detector for this tritium that we can put into a machine producing as much heat and pressure as the sun, and over many years will withstand the radiation equivalent of thousands of concentrated bananas. Turns out, the ideal material for this is diamond. Now, we think of diamond as big, blingy jewellery, but diamond has a myriad of fascinating properties which make it ideal for use in electronic devices, and I wish I had more time to tell you about all of these. We can also grow diamond in a lab, and this means that we can grow it to the exact dimensions we need for our electronic device. And really thin diamond films are ideal for radiation detectors. So how do they work? When tritium undergoes radioactive decay, that is, it loses the unstable bit that makes it radioactive, it releases an electron. And the more tritium there is, the more electrons are released, these red wavy lines on my diagram. My detector is made up of a thin diamond film with metal on either side that is then made up as part of an electronic circuit. And it measures the number of electrons that hit that thin diamond film. So we can work out how much radiation is present. The more electrons there are, the bigger the reading, so we know that there's tritium present, a lot more tritium present. But if we don't get a reading, we know that there's no electrons hitting the film and there's no tritium present. Now, all this sounds fairly simple. Take a piece of diamond, whack some metal on it, throw it into the corner near the miniature sun, and we'll know how much tritium there is. Simple. But if it was, it probably wouldn't be the topic of my PhD. There's still plenty that we don't know. We don't know how thin the diamond needs to be. We need the detector to be small so it fits into the circuitry, but we don't want it to break. We don't know what other molecules might be present. These molecules could also release electrons and tell us that there's radiation when there isn't. There's no tritium. And we don't know what metals can be used to make up the diamond detector sandwich. Just because diamond can withstand that much heat and pressure doesn't mean everything else can. But maybe, once I've worked all of this out, I'll be able to tell you how many bananas worth of radiation there is in a miniature sun somewhere in the Oxfordshire countryside. Thank you very much.
Last year, I was cycling home late at night when a badger ran out in front of me. It went straight into my front wheel, sending me flying over the handlebars. Luckily, we were both absolutely fine, but I did end up with deep cuts across my knuckles. You may be wondering why I'm telling you this. The first type of immune cell to be recruited to these cuts are neutrophils, and it's this cell type that is the focus of my research. Neutrophils are visible as the main components of pus, but they are far, far more important than that. They act as the body's first line of defense against invading pathogens. They circulate in our bloodstream until they are recruited to sites of infection or damaged tissues. Once there, they destroy invading bacteria using the mechanisms highlighted on the slide. Neutrophils consume bacteria in a process known as phagocytosis, release proteins from granules which target bacteria directly, and shoot out their DNA to form stringy nets which entangle pathogens. And neutrophils do all these things despite having an extremely short lifespan of less than 8 hours. And what this means is that our body produces neutrophils at a simply phenomenal rate. Neutrophils are produced from stem cells in our bone marrow. And the time that I've been talking, you will have each, hopefully, produced around 50 million neutrophils. I say hopefully because when neutrophil numbers drop, we're in real trouble. And this is especially true for cancer patients who have chemotherapy treatment. Now, chemotherapy disrupts the production of neutrophils. Without them circulating in the bloodstream, patients are prone to life-threatening infections. Although patients are given neutrophil transfusions, the short lifespan means that most of the transfused cells are already dead, reducing the efficiency of this approach. The short lifespan also complicates the study of the cell biology of neutrophils. To address this problem, my research aims to produce neutrophils in the lab. To do this, I isolate stem cells from blood samples. I then culture these stem cells in the lab, exposing them to factors that are present in the bone marrow. This forces stem cells to expand, but to ultimately mature into neutrophils. So I can now use this protocol as a model system to examine diseases in which neutrophil production is disrupted. A process that normally occurs in the bone marrow, so it's actually really difficult to study. I've also shown that lab-grown neutrophils possess all the necessary bacteria-killing mechanisms that I talked about earlier. This means that if we scale up this protocol, it's possible that we can use it therapeutically. Potentially, for cancer patients, these fresh, lab-grown, patient-matched neutrophils could be used as an alternative to transfusions. Thank you for listening.
I'm Robert Bickers. I'm Associate Pro-Vice Chancellor for Postgraduate Research, and I'm delighted to be able to announce the winner and runner-up. So here we are, 3 Minute Thesis 2020, organised by the Bristol Doctoral College. Gosh, well, this has been a surprise for all of us. It's online, and nobody knew this when they applied to join, and it's been a fantastic competition, and the competitors have really learned a lot on the way. As a judge, it's been really, really difficult to reach a conclusion. The standard of presentations was very high, really effective communication through slides, and some highly advanced dramatic skills, I think, in 1 or 2 cases, and a lot of really great potential in the projects that we've had explained to us in 3 minutes. We have a runner-up, Sophie Osborne, and in fact we have a winner, David Brigido. So congratulations to both of those.
I'm absolutely delighted to find out that I've been the runner-up of this year's 3 Minute Thesis competition. I originally applied for this in a very different world, pre-COVID, expecting to present to, I don't even know how many people. And for me, it was a personal challenge because I find presenting very difficult, and then being told that I was going to be doing it online in this completely new format. It was a challenge, but it was also kind of fun to get to do it like this, in such a different way to what I was expecting. I feel like the competition was really well run. I would absolutely recommend it to anybody, and thank you again to the judges for giving me the runner-up position.
I can't believe it, to be honest. I don't think it was possible, but I'm so happy. And obviously, I just want to thank my main supervisor, Benjamin Woods. He helped me a lot. And also my friends from the Access Department. They also gave me a lot of support and help. Well, I actually want to also thank my family and, well, and the persons that really care about me in any point, and persons who really care also. Well, I think that's it. And I had a lot of persons that were close to me all the time. And believe me, I think in coronavirus outbreak, if you're alone and you're facing a lot of problems here, it's very difficult to concentrate because there's no people to talk about. So it really helps to be in contact and online with a lot of people. So, well, thank you very much, and thanks a lot.
Thank you all for watching. Please vote for the People's Choice. And please make sure you do. Only once, please. And make sure you vote by 9 AM on Monday the 15th. 9 AM, Monday the 15th, People's Choice, 1 vote only. Thank you.